Okay, so this is all the kind of like cell type questions that could come up for your Unit 1 Applied Science exam. So the first question is asking you to label the parts of the cell. So we've got our cell membrane. Remember, our cell membrane is always the second kind of layer, and your cell wall is the first layer, and then you've got your cytoplasm in the middle. Okay, be careful of this because it says label A and B. You've got to be careful that you make sure to do it because that's too much you could have lost. Then it says complete the table. The nucleus, that contains DNA. The site of photosynthesis is the chloroplast, and then the cell wall gives the cell its structure. Okay, name one part of the algae cell that would not be present in an animal cell is the chloroplast. You could have also written the cell wall. Okay, they would have been the two big things that wouldn't have been present. Okay, again, this question, it says no labels are required, but I did just label it just to help you out a little bit. It says the diagrams below show an animal cell and the cell membrane of a plant cell. Complete the diagram. Now, the big thing about this is they've drawn the cell membrane, you making sure you put the cell wall outside. If you put the cell wall inside, you wouldn't have got the marks. Okay, I've then put the nucleus, the vacuole, chloroplast and the cytoplasm. It says no labels, but I just thought I'd label it just to get, it'd be good practice for you in terms of revision. What's the function of the cell membrane? Controls what exits and enters the cell. Then there are two diagrams, okay? Both are carrying out respiration. All of these are oxygen molecules, okay? Remember, the oxygen molecules want to go into your cells so that we can respire. Thinking about respiration, that is how we make energy, okay? So oxygen does want to go in the cells. Okay, if you look, there is in both of them, there is a higher concentration outside the cells than there is inside the cells. Okay, so there's more oxygen particles outside of the cells than there are inside the cells. Okay, there are then three questions. In cell A, the oxygen molecules move into the cell. Okay, in cell B, the oxygen molecules also move into the cell. Now be careful of that because that's kind of a tricky question in that you most probably think it's different, but it isn't. Okay, they both move in. Where would there be a greater net movement of oxygen would be cell B, okay, because there was less already in the cell, so there'll be a greater movement of them. Name the process by which the oxygen molecules move in is diffusion. Question number 3 then, another labelling of a cell. They've labelled the cell wall, look, it's the outside one, then your cell membrane is the inner one, okay? This bit in the middle is the vacuole. You can see the same kind of questions come up all the time. So chloroplasts are the site of photosynthesis. They are not present in an animal cell. Okay, mitochondria makes energy. They are present in both types of cells. Nucleus contains the chromosomes. Chromosomes, DNA. Okay, so be careful you don't fall down on that word. Yes, it's present in an animal cell. Another question, you can see it's exactly the same thing. So cell wall is this one on the outside here. Cytoplasm is all the little bits outside. You can see the little dots of the mitochondria in it, and the vacuole is that big bit in the middle. Okay, can you see how they've got cell wall, cell membrane? Okay, what's the function of the cell membrane? Controls what exits and enters the cell. Two structures that would not be present in an animal cell. So that's going to be A and C. Okay, A and C. Name the structure drawn which would be present in both an animal and yeast cell. A cell membrane. Okay then, so moving forward, the next bit then you'd have to use a ruler to measure the distance between there. I got 15 millimeters, you might have got something different, which is fine, depends on how the page is printed and what size the printed. Then to the magnification, times it by that number and it will give you an actual length. Okay, structure P is that vacuole in the middle. Then it says name the structure, the site of respiration. Okay, you know that respiration makes energy, so that should be nice and easy. Controls what exits and enters the cell, cell membrane. Chloroplast, the site of photosynthesis, and then nucleus contains chromosomes. Then it says state the meaning of diffusion, the movement of particles from an area of high concentration to an area of low concentration. Okay, then there is a lot of information. These questions come up all the time. I want you to be thinking about starch is a lot bigger. Okay, so when it's a lot bigger, it means that it won't fit through the viscin tubing. Okay, imagine the viscin tubing is like little lines like that, like your cell membrane. The starch is too big and it won't be able to fit through. Okay, whereas glucose is nice and small and it can fit through. Okay, so that's a reason as to why you don't see starch moving through the viscin tubing. Okay, state why starch does not appear in the test tube. Okay, starch is too big to fit through the viscin tubing wall. Suggest a temperature that would work for the fastest rate by circling your answer. Okay, you need to look at the table before, and I gave 100. Again, the definition of diffusion again. Movement of particles from a high concentration to a low concentration. Again, they've got this Viskin tube in prac set up. You can see the same types of questions come up over and over again. Again, another one, okay? This one's a little bit different in that it says what is meant by the term selectively permeable. Selectively permeable means only let certain particles through, not all particles, just certain particles, okay? Just talked about it because molecule A is far too big to go through, okay? State the process by which molecules pass through the Viskin tubing. This is actually diffusion because osmosis is the movement of water, so it's not actually water that's moving, it's the other molecules, so that is incorrect. Identify which molecules pass through the Viskin tubing. Molecule B, it is small enough to let through and fits the pores. You could have said something like A is too big. Explain why the contents of the tube turned blue-black. Okay, so inside the tube is the first thing they're asking. Okay, remember iodine is the test for starch, and starch will turn from like an orangey colour to a blue-black colour. Okay, so if you put, remember there's loads of starch within the Viskin tubing. Okay, if you keep that there, then the starch molecules will turn the iodine blue-black. Okay. The iodine is small enough to fit through the Viskin tubing, so it will be able to move between the Viskin tubing and the tube of water as well. Explain why the colour of the iodine solution in the boiling tube did not change, okay? Starch is too big to fit through. Remember, the Viskin tubing is tiny little pores. The starch is too big to fit through, so it will stay in the Viskin tubing, okay? Name one substance required for respiration that would pass into the cell. That is glucose, okay? Remember, the glucose molecules are really small, so they will fit through the tubes. Okay, starch stays in there. Iodine's the test for starch. There would be no starch out here, so the iodine would stay like an orangey colour. Then just plot on a graph. This was a weird one where it says join your points with a ruler. Okay, it's weird. That's unlikely to happen. But if it does, see there's a nice like set of instructions there. Okay, you've got 1 mark for doing the scale, 2 marks then for plotting the points, and then a final mark then. A final 1 mark then for plotting the points, okay? You then had to use your points to answer the questions on the next page, okay? So it's 6.2, it was at 20, wasn't it? So 20 and then down. From the graph opposite, which is on the previous slide, describe in details the effect of pH on the activity of amylase. Enzyme activity increases until pH 7.5, which is optimum, which is when it decreases. Calculate the difference, so find the value at 6.2, find the value at 7, and take them away from each other. Student did not keep the temperature constant during the investigation. Why did this prevent the investigation from being a fair test? Temperature can also affect enzyme activity. Really nice question there for 2 marks. Complete the diagram below to show the next stage of the reaction between the enzyme and the 2 simple molecules. They will attach the active site. This is known as lock and key. What would effect would boiling have? Okay, boiling would change the shape. Okay, boiling would change the shape of the active site, so these small molecules will no longer fit. They can't bond, you can't break them down. If you wanted to add like another stage here, it would kind of be like you get loads of small bits, okay, which is really important for digestion. We need the smallest bits as possible. Again, another graph, okay, again you had to plot an axis down the bottom, plot the points, okay, this one was one where you had to do it on your own, and again you had to extract information from the diagram. There's the marks for that. Okay, describe how enzyme activity changes. It increases until 40, it then starts to decrease. Another question again, where you were getting the information from the graph and taking them away from each other. Give the volumes of the boiled lipase solution of milk that would be used. Boiled lipase would be 1 and milk would be 5. Same volumes needed to ensure accuracy. Give a reason why there's no enzyme activity in the control tube. The lipase is boiled, so the enzyme is denatured. What does lipase act on? It is fat.